Hello, good evening everyone. Today we are going to start the nerve physiology orientation class and we are going to talk about the nerve injury and degeneration and regeneration of a nerve fiber. Now before that, we need to know about the classification of nerve fibers. Now nerve fibers, they are all not of the same type. Some nerve fibers are motor in function, some nerve fibers are sensory in function. So there are like two ways of classification that has been done. First, the scientist called Erlanger and Kaiser. What they done? They have classified both the sensory and motor nerves. Sensory nerve means which will sense the sensation like pain, temperature, touch, pressure. All those sensation will be carried by such group of nerve fiber which we call as the sensory nerves. Now motor nerves, these nerve fibers are designed and specialized in such a way that they go and innervate the structures which are for the motor function, for the movement, like the muscle of the limbs, the back, all those muscles. Okay. Now, the criteria for this classification, according to the Erlanger and Kaiser, are that they have taken into account the myelination. Okay. Then the diameter of the nerve fibers, because both myelination and diameter, what we have known, they are important in determining the speed, the velocity of the conduction of impulse. Then they have also taken into account the velocity, the velocity of the conduction. So if we write and make a column, the classification stands out to be. Like one thing about this Erlanger and Kaiser is that they used the alphabets like A, B and C to classify. Okay, now going by the classification, what are the classification? Now this is the fiber types, like whether they have classified it as A, B, C, that. Then we have the status of myelination, like how much a particular nerve fiber is myelinated. Third, we have the diameter in microns. Fourth is the velocity of conductance, which is like meter per second. And third is their function in context to location. Like where are they located and what is their function in context to that one. So going by the first type is A. Now this A has again been subdivided, like further classified into A alpha, A beta, A gamma and A delta. So this A alpha, this is myelinated. It is not only myelinated, it is very heavily myelinated, very thickly myelinated. Now the diameter of this is about 13 to 50 microns, 50 micron, it can vary. And the velocity, since it is heavily myelinated, the velocity, the speed at which the impulse is conducted is very fast, because thin myelination will let the saltatory conduction happen, that we discussed in the previous class, right? So the velocity here is about 70 to 120 meter per second. Now, what is the main function of this nerve fiber? What type of impulse will it like transmit? It is mainly concerned with the proprioception. Proprioception means the feeling of the position. Like the person is able to feel where the joints are. The feeling of the position while doing a function is called the proprioception. So this roughly comes under the criteria of pressure. So we can remember like A alpha, it is for sensing the pressure. Second one is the A beta. Now this is also myelinated, very good myelinated, but its diameter is smaller. It is about like 10 to 15 microns, and the velocity is much slower. It is like 25 to 40. Now what it senses? It senses temperature and pain. It senses the temperature and pain. Third, according to this Erlanger and Kaiser, this A gamma, this is also myelinated, and it has even smaller diameter, which is like 7 to 12 or maybe 10 microns also, and the velocity of conduction is very small. It is like 7 to 13. That's all how much it is. Now here, this is not a sensory fiber. This is a motor fiber. So this is like, its function is motor supply to the muscle spindles. Okay. Next we have is the A delta. The A delta, it is myelinated obviously, but not heavily myelinated, and its diameter is further less. It is like about 3 to 5 micron, and the velocity is even less, which is 2 to 4. And here it senses again temperature and pressure. Now these are the good myelinated or heavily myelinated nerve fibers. There are other classifications also, like the B and the C. Now B, this is like myelinated sparsely. Okay, it is myelinated sparsely, and the diameter is very less. It is about 1 to 3, and the velocity of conduction is also very slow. It is about 2 to 2.5, and it also is a motor supply. So its function is the supply, and it is preganglionic sympathetic fibers. Okay. C, this is unmyelinated. Its diameter is very small, 0.2 to 1, and velocity conduction is 0.5 to 1. And this one also senses, like, it senses temperature and pressure, and also it has a branch which is the postganglionic sympathetic fiber. So this is the classification according to the Erlanger and the Kaisers. Now Erlanger and Kaisers, they have the classification based on the mixed nerve, like the both sensory nerve as well as the motor nerve. Whereas Lloyd has taken this classification, and the same nerve fiber he has classified Lloyd and Hunt, both of them, like especially Lloyd, what they have done, they have classification is only for the sensory nerve fibers. They have not classified the motor nerve fiber. So all this nerve fiber which has got sensory function, they have been classified. So here A alpha has been classified as 1A depending on its origin. When this A alpha fiber, which is myelinated, its origin is in the muscle spindle, then it is called as the Roman A, and A alpha, it is Roman B, like Roman 2B, whose origin is in the Golgi tendon. Okay, these are the classification. Now, A beta has been classified as 2, and what it senses? We have already seen it is temperature and pain. These are the two things that it senses. Now, A gamma, if you remember, it is like it has a motor supply. So, any nerve fiber that does only the motor supply, that has not been taken into classification by the Lloyd and Hunt. So, A alpha we don't find it. Whereas A delta is like 3, which again senses what? It senses the temperature and pressure. Okay, pressure in form of pain also you can say. And here B, which is the preganglionic sympathetic fiber, that has not been taken into account, whereas C is classified as 4, and what does it sense? It also senses temperature and pressure. So this is the both way of classification of the nerve fiber. Okay, coming to the next point, that is the nerve injury. Now, what is a nerve injury? When does a nerve gets injured? Say for example, if a person suffers from any trauma, like there has been a fracture of the hand, or there has been a mob lynching in which his bones got broken, any kind of trauma, it causes injury to the nerve. Now, injury to the nerve always does not mean that it is very severe, very extreme. It can happen in the lighter severity also. So, nerve injury, depending on the severity, has been classified by Seddon and Sunderland. Now, before understanding this one, just look at the hierarchy, like how a nerve fiber lies. Now, it is closed by various fascicles covered by the connective tissues. Now, the outermost tough sheath, that is covering a bunch of nerve fiber, it is called as the epineurium. Now, inside this epineurium, there are like fascicles. There are this fascicles. Okay, fascicle means a bundle or bunch of the nerve fibers. So these fascicles are covered by a tough covering, which is called as the perineurium. which is called as the perineurium. Okay, my chat box, excuse me, I can't see anything in the chat box. Hold on, I have to just refresh the chat box. So I can see only one chat which is called, okay, Hidaya, pharmacology. No, I don't teach pharmacology. I teach only physiology. So any chat other than that, please let me know because I can't see any chat. It's not visible. So the tough covering, that is the connective tissue covering that is enclosing the fascicle is the perineurium. Now inside perineurium, there are like nerve fibers. There are this single nerve fibers as we studied, which is having the axon, and outside the axon is a myelin sheath, which again is protected by a connective tissue, which is called the endo. Yeah, which is called the endoneurium. So this is the hierarchy. So whenever there is any injury to the nerve, it means it will be called an injury if there has been damage to any of this structure like this epineurium, perineurium, endoneurium, or even if the axon or the myelin sheath has been disturbed. Okay, so there are basic three type of classification according to the Sunderland classification. According to Sunderland's classification, nerve injuries are of following types. Number one is neuropraxia. Now, neuropraxia, it means when the nerves gets compressed. Here, the nerve fibers get compressed. Suppose a person is having a fight or a bout of argument, and in that bout, someone punches a person in the neck. So, what do we see? There is a swelling in the neck, right? This type of a thing. Or if a person sleeps with on one hand folded, okay, in recumbent position for a prolonged period of time, then what happens that time also like there is compression. So these all causes neuropraxia. So what happens in neuropraxia is not actually a very serious condition. Here only the myelin sheath, we have seen this is, if this is the axon, so this is our myelin sheath, right? So in case of neuropraxia, what happens is that the axon is not at all disturbed, only there may be some like wearing of, there may be some like wearing of slight injury to the myelin sheath, some injury to the myelin sheath only. Okay, injury only. Now here the recovery rate is very high. It recovers within minutes. That's all it happens. And there is temporary stoppage of conduction of impulse beyond the area of injury. That's all it happens. Okay. The second type is the second classification. First is the neuropraxia. The second one is the axonotmesis. Is the Exonomesis. Now in exonomesis, what happens? This is called as the crush injury. It is more severe than the neuropraxia. Here, the injury is such that the nerve fiber kind of gets crushed. It is like crushed. So, here, the myelin sheath and the axon gets damaged. So, two things happen. The myelin sheath also gets damaged, and the axon inside that also gets damaged. So, if we draw it, it may be something like this. Okay. Exonomesis. So, it is like this. Myelin sheath are damaged. Okay. Not only that, the axons are also not intact. This is exonomesis as we see. Okay. So another characteristics of this axonomesis is that Valerian degeneration occurs. Now, what is Valerian degeneration? We will come right after it. So, here in exonomesis, Valerian degeneration occurs, and it takes days like 2 weeks to recover. Depending on how severe it is, it may take from a few days to like maybe a week or so to recover. Third is the neurodemesis. Okay, neurodemesis. Now here what happens in neurodemesis, it is a transaction injury. Example, if a person has like a cut injury, a sharp cut of the limb, like through a glass, laceration, or like through a saw, one arm, one limb, one part has been totally transacted, totally cut, then it is called neurodemesis. So here, the whole constitution, like the epineurium, perineurium, and endoneurium, may be this time. This is what it may happen. This is a transaction injury or like total sharp cut injury. In this case, surgical intervention, like a surgery is needed to correct this kind of injury. Surgical intervention is needed. Okay. And also Valerian degeneration occurs. Now, it takes very long time, that is maybe months to recover. Okay, these are the type of the nerve injury classification according to the SID. Now, this one has been further taken and classified by Sunderland. Now, Sunderland has proposed a different type of classification which is now widely in use. He has classified the injury as per degree depending on the severity, like how severe it is. That is the degree. So here he has used like 5 degrees to classify the injury. Like first degree. First degree of the injury as per Sunderland Classification is the neuropraxia. Is the neuropraxia. Same thing. It is totally same like neuropraxia, where only the myelin sheath gets worn off, right? And it is the least severe form of injury. Now, second degree is neuropraxia, and like disruption of axon. Disruption of axon. Here, the endo, epi, and perineurium are all intact. And in second degree, here the Valerian degeneration occurs. Here the Valerian degeneration occurs. Third degree is like axonotmesis plus disruption of the endothelium. But the epi and perineurium are intact. So here what happens, it is the same event like axonotmesis, like the myelin sheath has got worn, the axon has got distangled or like has got damaged, the exoplasm has got damaged, but also the endoneurium, the innermost layer. What is the innermost layer? This is the layer that is surrounding an axon, right? The layer that is surrounding an axon that gets damaged. That is the endoneurium. But the peri and the epineurium, they are intact. So the hierarchy, the organization of the nerve fiber is still maintained. So here also Valerian degeneration occurs. Okay, this is the third degree. Coming to the fourth degree. Fourth degree, what is it? It is again axonotmesis plus the endoneurium and perineurium. Perineurium means the second layer of the connective tissue that is covering the fascicle, a bunch of nerve fibers, right? So axonotmesis plus endoneurium and perineurium gets affected. Okay. Valerian degeneration is seen and surgical correction is mandatory. Like surgical intervention is needed no matter what. Now last come the Rakesh Prajapati. Good evening to you also. So the last one is the fifth degree, the most severe type of nerve injury. Here, it is out and out neuron meiosis, like total disentangling of all connective tissue layers surrounding the nerve. That is the endoneurium, the perineurium, and epineurium all get distangled. Okay, the whole hierarchy is destroyed. Here, surgical intervention is needed, and the recovery is also not guaranteed. So, these are the classification of the nerve injury. Now, coming to the degeneration and regeneration of a nerve fiber. So, first we start with the degeneration of a nerve fiber. Now, degeneration of the nerve fiber, we know what is the criteria for a nerve fiber to get degenerated. Whenever there is a trauma or whenever there is an injury and the nerve gets compressed or crushed, or there is any kind of even ischemic condition that is blocking the supply of oxygen, then also there is nerve injury or damage to the nerve. So, what are the criteria for a nerve injury? After following a nerve injury, what are the criteria for degeneration? So, criteria for degeneration, like ischemia. Okay, there can be ischemic condition, lack of oxygen, crush, compression. All these are the conditions which can lead to the degeneration of a nerve fiber. So, what are the stages in degeneration of a nerve fiber? Drawing the neuron, a typical neuron once again. So, say this is the axon here, this is our axon, and this is the like synaptic terminal. This is a cell body, and we have the myelin sheath. This is the myelin sheath. Let us imagine there has been an injury to this nerve due to some crush injury or damage to the injury due to some reason. At this point, there has been a damage at the level of this point. Here, there has been, this is the site of injury. Okay, now just the point above the site of injury and below the site of injury, there will be certain physical as well as certain chemical changes. Now, the area which is above the site of injury, which is above the site of injury, that is upward. This is upward. This is called as the proximal site, and the site which is just below the site of injury or the point of injury, this is called as the distal site. So when the degeneration changes happen, like degeneration in the physical and the chemical constituency or the properties of the nerve fiber, when this changes happen in the proximal it happens in the opposite direction. Opposite direction means what? This is the site of the injury. So from the site of the injury, the degeneration is happening backward, right? It is happening backward, like from the site to the cell body. So this is called as the retrograde degeneration. Means the degenerative changes or the destructive changes. Degenerative change is also known as the destructive change. This is happening in the retrograde manner. This is happening in the retrograde manner, in the opposite direction. So we call it as the retrograde injury, as the retrograde degeneration. Whereas in the distal site, the changes, the physical and the chemical changes that is happening after injury, this is like away from the site of injury towards the axon terminal or synaptic terminal, that region. So it is in like from upward to downward, upward to downward. We call it anterograde degeneration. Now, one thing about the anterograde degeneration, in the distal site, this anterograde degeneration is the degenerative changes that we will study now. This is what we call as the Valerian degeneration. So, what is a Valerian degeneration? This is the destructive means the change that is destroying the nerve fiber further after the injury. Destructive changes in a nerve fiber following an injury. Like after an injury, what are the destructive changes that is destroying, that is damaging the nerve fiber further? So what are they? So it starts with the melting or degeneration of myelin sheath. Now coming to myelin sheath, what did we study about the myelin sheath? Myelin sheath, it has more lipid and it has more lipid and less protein. This is the only membrane that is surrounding a cell which has more lipid and less protein. So melting or degeneration of the myelin sheath into fat droplets, because it is mainly made up of lipids, it is made up of fat droplets. And then what happens inside the myelin sheath? What do we have? We have the axon and axoplasm. So dissociation of the exoplasm from the axon. Now, what happens after this? This droplets that is formed from the myelin sheath and all, the macrophages and Schwann cells scavenges the melted myelin sheath. So, if we draw it like if this is the neuron, and there has been a damage here. So when the Valerian degeneration sets, Valerian degeneration happens, what will happen? The myelin sheath, it is dissociated. They have dissociated. Now once they have dissociated, what follows? I'm just drawing this part alone. What follows? There is distangling of the axons also, where the myelin sheath on its sides have precipitated as oil droplets. Now, this oil droplets will be scavenged upon by this macrophages and Schwann cell. They will clear up this droplets which is formed due to melting of the myelin sheath. These are the degenerative change. And one thing to remember, degenerative changes or Valerian degeneration occurs depending on the severity 12 to 24 hours after injury. So this is the changes in the Valerian degeneration. So when this is happening, obviously what will happen in the terminal area? That this axon terminal, there will be dissociation of the axon terminal, okay, and the muscle that the nerve fiber is innervating will become atrophied. Like if the muscle that this nerve fiber was innervating, if that muscle was say like this, after degeneration and loss of innervation, because the nerve fiber has degenerated, the muscle will shrink in size. It will shrink in size. Okay. Now this is what has happened in the distal site, the Valerian degeneration. Now there will be some changes in the proximal site also. So the changes in the degenerative changes in the proximal site, like the site which is above the site of injury. So what is there? The changes that we studied about the myelin sheath and the axon, that will happen there also. Additionally, the cell body, say it was like this. Now, here there will be drawing of water, like more water will enter the cell body and it will swell up. It will become like round and it will like be roundish and it will swell up a lot. There will be water that has entered and there will be flattening the corners from where the dendrites are there. It will be much more flattened. So there will be like swelling up of the cell body. Now, after swelling up of the cell body, there will be chromatolysis. We know that a neuron, what does it have? It has endoplasmic reticulum, right? which we called as nasal. And under the microscope, they appear as purplish, right? So this cell body, it will undergo chromatolysis. So what will happen in chromatolysis? Here, the swelled up cell body, which has drawn a lot of water, there will be like bursting. The nasal granules will just burst and become a dust like particle. The granules will become dust like particle. Okay. So these are the changes in the cell body. Additionally, the organelles like mitochondria, mitochondria, Golgi apparatus, okay, organelles like Golgi apparatus, mitochondria will disintegrate, So in the proximal site, first the water will move from the ECF and enter into the cell body. So there will be increase in the volume. There will be swelling up in the volume. Then there will be like nasal granule burst, which is chromatolysis. And what is nasal granule? Nasal granules, just all of us we know it, just for the mention, nasal granules or nasal particles or nasal bodies are the rough endoplasmic reticulum in a nerve fiber cell body. Okay, this is what it is. After that, the organelles like the Golgi body, mitochondria will disintegrate. Now, what happens to the nucleus? Does the nucleus like goes out? Is there any pyknosis or any kind of disintegration to the nucleus? No. The nucleus here will be pushed to the periphery. Usually, nucleus is in the center. Here, the nucleus will be pushed to the periphery. So, these are the degenerative changes. Okay. This will happen, it will start within 12 hours to 24 hours post nerve injury. Now, after that, like after 36 to 48 hours, there will be again reparative changes. So, after 36 to 72 or say 48 hours, the reparative changes will begin. Okay. Now, these changes are the regenerative changes. So just before the regeneration starts, what happens? There is destruction of the contact between the nerve and the muscle. The nerve fiber that was innervating the muscle, that contact is lost and the muscle has become smaller in size, has become atrophied in size. Now, after that, there will be reparative or regenerative changes, means the nerve fiber will again be laid back to almost normal. Now, for that also criteria are there. We will study after this regenerative changes. So, it again starts with the distal part. So, from the distal part, what happens in the in the distal part, what happens is that this is the axon. So from that part, there are like formation or laying out of the myofibril. There are like numerous myofibril that are like given out. About 50 to 100 myofibril sprouts. About 50 to 100 myofibril sprouts are given out from the damaged axon on the distal side. This is how it starts. But out of this 50 or 100 myofibril, only one myofibril can successfully invaginate the axon. Because once the axon was left, the neurolemma was a hollow tube. So, out of so many myofibril, only one myofibril can actually go and invaginate. Only one myofibril invaginates the neurolemma. It invaginates the neurolemma, and then again the myelin sheath is laid by the Schwann cell. Then myelin sheath is laid once again by the Schwann cell. This is what it happens in the distal area. Now, on the proximal part, on the proximal side, what happens in the proximal side? Pretty much the same thing happens. Again, the laying of the cell body of the neuron happens. Now, the cell body of the neuron, it assumes the normal shape and the nucleus comes to the center. Once again, there is appearance of the mitochondria and also the Golgi apparatus, and also there is like the nasal granules which is dispersed in the cell body. So the steps we can write what happens actually here. The nucleus once again comes to the center of the cell body. Okay, it occupies the central position, and organelles like mitochondria, Golgi body reappears, and there was that swelling of the cell body due to incoming of the water, right? The excess water that entered the cell body leaves the cell. Okay, so this is how the regeneration happens. Now we have spoken about the degeneration, regeneration. Here comes the important concept. Does all nerve fiber after getting damaged or destroyed, they come back to normal? They're all again like laid back to the normal orientation? No. it is only the nerves of the peripheral nervous system, that can be regenerated, not the central nervous system, nerve fiber. Nerve fibers. Now, what is the reason behind that? Why it can't happen? Why this is not possible? The reason is why so? It is so because in peripheral nervous system, the myelin sheath is laid by the Schwann cells. Myelin sheath is laid by the Schwann cells. But in CNS, it is the oligodendrocytes that lay the myelin sheath. So, after injury, the Schwann cells can relay the myelin sheath. It can relay the myelin sheath, but oligodendrocytes cannot. Oligodendrocyte cannot relay the myelin sheath. This is one reason. Another reason, which is thought to be the reason for not being able to regenerate the nerve fiber in the central nervous system. Another reason is that after injury in central nervous system, there are scar formation. Okay, there is inflammation, and also there is release of cytokines. All these makes laying of myelin sheath difficult. This is one established fact. Some other theories are also being proposed, though they are not proved. So, what are the other theories? Other theories, these are not, I repeat, these are not confirmed. It is not proved, but still there are these theories. It is thought that presence of NOGA receptor NT75 and then like MAG, OMG, these are all the myelin sheath proteins that are present in the myelin sheath. So it is seen that presence of this protein also inhibits the myelin sheath lay. Okay, so these are the reasons. So this is a theory, it has not been proved. So these are the possible reasons why there is no regeneration of the central nervous system nerve fiber. Okay. For this regeneration, there are certain factors which are essential. There are certain supporting factors, like certain proteins which are needed. So such proteins are the neurotrophins. Neurotrophins. Now, what are neurotrophins? Neurotrophins, these are proteins that are produced by astrocytes. Now, astrocytes, these are like star-shaped nerve cell which does a supporting role. Now, this neurotrophins enter the nerve fibers by endocytosis. So, by the process of endocytosis, this neurotrophin are entering a neuron. Okay. What are the types of the neurotrophins? A few names for us to remember. Like for example, nerve growth factor. Then we have the neurotrophin 3, neurotrophin 4 and 5. Then we have brain derived neurotropic factor. Okay, neurotropic factor. So these are the examples of a few neurotrophins. Now, these neurotrophins, they use a particular receptor. They use a particular receptor to get inside the cell. That gets inside the cell. So, which glycoprotein facilitate endocytosis? This is what actually we are talking. See this. This is like this receptors. This is a protein in nature which is present in the cell membrane. So this growth factor, which are protein, which are hormone in nature, protein macromolecule, they bind on those receptor, and then by the process of endocytosis, they come inside. They get pinched inside. So the receptors are like for nerve growth factor, the receptor is tyrosine kinase A. For neurotrophin 3, it is tyrosine kinase B and tyrosine kinase C. For neurotrophin 4 and 5, it is again tyrosine kinase B and brain-derived neurotropic factor, it is again tyrosine kinase B. Now, various clinical conditions are seen Various clinical conditions are seen whenever this neurotropin is deficit or they are not functioning properly. Okay, so what are the deficit conditions? Okay, in context to the neurotropins. So functions of the neurotropins. First is the nerve growth factor. Now this is needed by this is used by the cholinergic and sympathetic neurons. So for the growth and survival of the neuron which is sympathetic in nature and cholinergic in nature. Cholinergic in nature means it secretes acetylcholine. So this nerve growth factor is needed for the growth and survival of the cholinergic and sympathetic neuron. Now deficiency of nerve growth factor leads to immunosympathectomy. Now, this is a condition that destroys the sympathetic nerve fiber. So, whenever there is any antibody that is raised against the nerve growth factor, it destroys the nerve growth factor, making it deficient, and this deficiency leads to immunosympathectomy. Second nerve growth factor that we have is the neurotropin 4 and 5. It is needed by the hair follicle cells for its growth and survival. Another one, as mentioned, neurotropins 3. It is needed by the mechanoreceptor of the skin and proprioceptors for their growth and nourishment. Now, brain-derived neurotropic factor or nerve factor. This is needed for the vestibular organs. Okay. Then we have other neurotropins like glial cell derived neurotropins. They are needed for the spinal motor neurons, ciliary derived neurotropins. Now, these neurotropins, they are needed for the spinal cord neurons. So, these are different or various neurotropic factor which is needed in different area for the growth and development and nourishment of the nerve fiber. Now, a small clinical condition for our concept knowledge, for our conception, a small clinical condition that is attached to the deficiency of this neurotrophic. Like, for example, it is seen that in clinically depressed person, the brain-derived neurotrophic factor or tyrosine kinase B, which is the receptor for brain-derived nerve factor or neurotrophic factor to bind, is low. So, it is seen that in a person who is suffering from clinical depression, either the brain-derived neurotrophic factor or the tyrosine B receptor, their concentration or their quantity is not correct. It is below normal. Okay, this is a clinical condition related to that. So, we have understood about the nerve fiber, the classification, degeneration, regeneration, and the role of neurotrophin in the regeneration, growth, and survival. So, Monday, next class, we will start with the essentials of muscle physiology. Okay, in the essentials of muscle physiology, we will understand the skeletal muscle structure, its function, which is of course contractility, few points to that, sarcomere, sarcotubular system, and then the excitation, contraction, coupling. Okay, so see you all on Monday. Till then, bye.